of research article I'd like to introduce is N-cadherin expression level modulates integrin mediate polarity and strongly impacts on the speed and directionality of glial cell migration from Journal of Cell Science. Cell surface adhesion molecules are the principal mediators of interaction between cells and are thus likely to be key players in tumor spreading. Generally, when loss of N-cadherin is observed, most often associated with N-cadherin is upregulated, which has a pro-migratory effect. In gliomas migration, the role of N-cadherin is still unknown. This study group have recently shown that N-cadherin is a key regulator in the polarity of non-migrating astrocytes. As loss of cell polarity is a common characteristic of tumor cells, they wondered whether alteration of N-cadherin levels in gliomas impacts on polarity and migration, a fundamentally formalized process. To investigate how N-cadherin mediated junctions could modulate cell migration, they used an in vitro Undeling assay that induced a collective directed migration. They analyzed N-cadherin levels in normal and glioma samples by Western blot. Comparing normal brain samples and high-grade glioma showed a significant twofold decrease in N-cadherin expression in glioblastoma, as you can see in Figure 1. Collectively, N-cadherin levels were variable but generally lower in tumor cells than in control astrocytes. That is, N-cadherin levels and adherin junctions are frequently downregulated in high-grade gliomas. Next result shows that decreased N-cadherin expression in primary astrocytes and glioma cells lead to a faster and less directed migration. From in vitro Undeling assay, wounding of a monolayer of primary red astrocytes induces a slow, cohesive, and directed migration. Trajectories of U373 and U138 glioma cells were longer and less directed than those of normal astrocytes. In contrast to normal astrocytes, especially glioma cells often escaped the monolayer and migrated as individual cells. From this data, they suggest that N-cadherin participates in the regulation of the speed and direction of glial cell migration, and this might ultimately strongly influence the invasive behavior of glioblastoma. Figure 3 presents immunofluorescence for anti-alpha tubulin and phalloidin stainings 8 hours after scratching. This shows the organization of microtubules and actin network in control astrocytes. N-cadherin depleted astrocytes, U373 and U138 glioma cells. Directed astrocytes migration is associated with the formation of an elongated protrusion in the direction of migration. On the other hand, in glioma cell line, the actin and microtubule network did not show a polarized organization similar to N-cadherin depleted astrocytes. They also observed the relative position of the centrosome and the nucleus to measure cell polarity. Level of expression of N-cadherin is a crucial parameter in the control of polarity in normal astrocytes and in glioma cells. They derived that N-cadherin is required for astrocyte polarization during migration. In order to determine whether N-cadherin reduced expression could influence cell matrix adhesion, they first examined the distribution of focal adhesions in basal conditions without any scratch. Non-fluent glioma cells had a significant higher number of focal adhesions than normal astrocytes, and that the mean size of this focal adhesion was smaller in accordance with depletion of N-cadherin. That is, N-cadherin levels influence the formation of focal adhesions in non-fluent cells. When confluent, control astrocytes were strongly adhesive with one another, and few focal adhesions were detectable. Numerous focal adhesions were visible in glioma cells as well as in confluent cell N-cadherin depleted astrocytes. Numerous focal adhesions were visible in glioma cells as well as in confluent N-cadherin depleted astrocytes. 1 hour after wounding, focal adhesion accumulated at the front edge of the cells and were polarized in the direction of the wound. Reduction of N-cadherin levels and inhibition of N-cadherin mediated contacts strongly affect the distribution of focal adhesions and prevent wound-induced formation of focal adhesion at the wound edge of the cells from these findings. N-cadherin is involved in the decreases of SERC inhibitory phosphorylation of tyrosine 529 in response to the wound downstream of integrin. Activation of SERC-like kinases is required for polarized migration of astrocytes. SERC tyrosine 529 phosphorylation was strongly decreased during the first hours following wounding of the control astrocytes. Decreased phosphorylation was associated with focal adhesion formation and signaling. Phosphorylated SERC tyrosine 529 was found in the most focal adhesions, irrespective of the wound orientation. N-cadherin-mediated contacts are required to restrict integrin-mediated signaling in time and space. Upon wounding, the raw guanine nucleotide exchange factor 7, which is called beta-fix, is recruited to the wound edge of the astrocytes, where it induced CDC42 and route recruitment and activates CDC42 to promote cell polarization and migration. Cell treatment with a cyclic RGD significantly decreased beta-fix recruitment to the leading edge. It consequently affected integrin-mediated beta-fix recruitment at the cell leading edge. In other words, N-cadherin-mediated contacts are required for integrin-mediated recruitment of beta-fix at the leading edge. With previous data, they suggest that wound-induced CDC42 for polarized activation and CDC42-dependent polarity pathway are altered in N-cadherin-depleted astrocytes and glioma cell lines. Upon wounding, CDC42 was recruited to the cell's leading edge of control astrocytes. In migrating U138 and U373 cells, CDC42 was still present on intracellular membranes, but was not enriched at the leading edge of the cells. Scratch-induced activation of CDC42 was dramatically reduced in U138 and U373 cells, as well as in N-cadherin-depleted astrocytes. N-cadherin is required for integrin and beta-fix mediated activation and recruitment of CDC42 at the leading edge of the migrating astrocytes. FAR6 and PK3-zeta shows an enriched cytoplasmic distribution at the leading edge of control migrating astrocytes. In N-cadherin-depleted astrocytes, U373 and U138 cells, the percentage of cells exhibiting FAR6 or PKC-zeta immune fluorescence at the leading edge was significantly decreased. N-cadherin mediated intercellular contacts control the integrin-dependent polarity pathway that regulates centrosome positioning and directed astrocytes migration. At last, they expressed a siRNA-resistant GFP-tagged N-cadherin construct in N-cadherin-depleted cells and glioma cell lines. Nucleopection of the GFP vector with control or N-cadherin siRNA had no effects on centrosome reorientation. Expression of GFP N-cadherin in N-cadherin-deflated astrocytes rescued centrosome reorientation, further demonstrating the role of N-cadherin in cell polarity. This result indicates that a decrease of N-cadherin expression is a major event perturbing polarity during glioma cell migration. N-cadherin expressing glioma cells migrated in a limited space area in comparison with their control counterparts. Interestingly, increased N-cadherin expression in glioma cell lines strongly reduced their mean velocity. N-cadherin acts as a brake to cell migration, and that the decreased expression of N-cadherin participated in the abnormal migration of glioma cells. Consequently, rescuing N-cadherin expression restored cell polarity and inhibits migration. This discussion section. First, this study suggests the role of intercellular adhesion molecule in the migration behavior of glioma cells. In the central nervous system, cells do not express E-cadherin but mainly N-cadherin, and no switch between cadherin has been described in the case of malignant gliomas, except in some specific cases. N-cadherin expression is variable but generally diminished in high-grade gliomas compared with the normal primary glial cell cultures. Using a siRNA approach as well as rescue experiments, N-cadherin is involved in the control of astrocytes and glioma cell velocity. Loss of N-cadherin without the re-expression of other cadherin should mechanically favor tumor cells migration away from the initial tumor by loosening cell-cell junction. In addition, they proposed that cadherin integrin interplay in the control of glial cells migration and polarization. In migraine cells, loss of N-cadherin also dramatically perturbed cell polarity and directed migration. Increased turnovers of focal adhesions might thus be responsible for the effect of N-cadherin on cell migration, promoting a faster mesenchymal-like migration in a wound healing assay. Downregulation of N-cadherin in glioma cells might induce a decrease in intercellular contacts in favor of more dynamic and less spatially regulated adhesion to the extracellular matrix, resulting in the abnormal migratory behavior of these cells. In conclusion, adherent junction proteins are now considered as key players in tumor progressions in different types of cancer. Creating N-cadherin depleted astrocyte by using a siRNA approach constitutes a good model to investigate the involvement of N-cadherin in glioma cell migration. The levels of N-cadherin strongly impacts on glial cell polarity and promote a non-directed migration. The decrease in the level of N-cadherin participate in the release of a migration brake caused by adhesive interaction with the neighboring cells and probably contributes to the increased cell velocity observed in glioma cells. Together, they conclude downregulation of N-cadherin as an impact event favoring the acquisition of the invasion property of glioma. This is the end of my presentation. Thank you.